Hello everyone. In this class you will learn about cyanobacteria, which is formerly known as blue-green algae. So let us see what are the content we are going to deal with. We start with the habitat of cyanobacteria, followed by the evolution of cyanobacteria, then the structure, pigmentation, and finally the endosymbiosis process. Cyanobacteria belongs to the division Cynophyta. Cyno, the word means blue. Bacteria, the word means to acknowledge that they are more closely related to prokaryotic bacteria than eukaryotic algae. Let's have a look on habitat of cyanobacteria. Cyanobacteria can be found in almost every conceivable environment, that is from ocean to freshwater, to bare rocks to soil. They can occur as planktonic cells or form of phototrophic biofilms in freshwater and also in marine environment. They occur in damp soil or even temporarily in the moistened rock in desert. Few are endosymbiotic in lichens and in plants and various protists or sponges, and they provide energy for the host. So let us see the evolution of cyanobacteria. Cyanobacteria are considered as the first organism to release oxygen, because we know that when life originated, the atmosphere was reducing. There was no molecular oxygen, and most of the cells which survived before that were unicellular and utilizing some other sources for obtaining their nourishment. Cyanobacteria used solar energy with the help of pigments, and they use water as one of the raw material. So from water, hydrogen was used by them and oxygen was given out as the byproduct. So they are believed to be the organism which converted reducing atmosphere into oxidizing by the process of photosynthesis. This credit goes to cyanobacteria. They are slightly differ from eubacteria by certain extra structures. Now, when we saw that they are performing photosynthesis, that means they have to have two photosystem pigment. The one pigment is chlorophyll A, and this pigment is present inside the membrane. So they must have a membrane bound structure. Some of the cyanobacteria can fix nitrogen. For fixing nitrogen, the enzyme which is required is nitrogenase. The enzyme nitrogenase works in anaerobic condition. So now, when it is working on anaerobic condition, this particular cyanobacteria is producing oxygen. Then how do they create the anaerobic condition? In some cyanobacteria, nitrogen fixation takes place in special thick wall cells, which is known as heterocyst. If we take one of the genus in cyanobacteria, that is nostoc, it has a filamentous structure and some cells are thick wall cells. They are known as heterocyst. In the heterocyst, the nitrogen fixation takes place with the help of an enzyme nitrogenase in absence of oxygen. If we talk about Oscillatoria, it is a filamentous algae and we cannot find any heterocyst in Oscillatoria. So it is not necessary that heterocyst must be present in all cyanobacteria. Now let us see the structure of cyanobacteria. They have peptidoglycan cell wall which is multi-layered. Around this there is a gelatinous capsule, that is jelly-like material is present on the outer cell wall. Inside the cell wall is cytoplasm, which is divided into two zones. The outer zone is where there are membranous structure present. This structure is called as thylakoids. So these structures are not in the form of stacks, that is, not in the form of grana. Now let's see the pigments in cyanobacteria. They have pigments like chlorophyll A, xanthophylls, allophycobilins, carotenoids, phycoerythrins and phycocyanin. Chlorophyll B is absent. Phycocyanin absorbs green-yellow light in the wavelength of 615 nanometer to 620 nanometer maximum. Allophycobilins emit red light with wavelength of 650 to 660 nanometer maximum. Phycoerythrin absorbs green light with the wavelength ranging from 495 to 570 nanometer. Because the thylakoids with the pigments are present in the outer surface starts to appear colored. So that's why the outer surface are called as chromoplasm. The inner cytoplasm material is clear because there is no pigment here. This part is known as endoplasm. In the endoplasm is the circular genetic material, the DNA or the nucleoid. Here there are ribosomes also, which is 70S type. Now this is the difference. In typical eubacteria, there is a peptidoglycan, cell wall, plasma membrane is also present. Cytoplasm is uniformly distributed. No thylakoid is present in the eubacteria, so no pigment is produced. But here, in case of cyanobacteria, there is an additional thylakoids are present in outer surface. Here, in prokaryotes, they don't have cell organelles. The same here with the cyanobacteria, they also don't have any cell organelles like chloroplast, mitochondria, Golgi body, endoplasmic reticulum, etc. The cyanobacteria perform photosynthesis because of the presence of the pigment, and the bacteria cannot perform photosynthesis because of the absence of thylakoids and pigments. And cyanobacteria helps in nitrogen fixation, and they are also found in the symbiotic association with other organisms. Example, bryophyte. Class Anthoxerox, it is found endophytically in this bryophytic thallus. In this slide, we will see the origin of algal diversity from the prokaryotes. How come the primitive bacteria-like structure have obtained their pigments? Scientists believe that these cells, they had endosymbiotic organisms. That means probably they were like simple prokaryotes and they had a endosymbiont. That is, they had another bacteria which was living outside, which was smaller in size, was taken in. So the bigger bacteria had the smaller organism, and it is believed that the smallest membranous organism had the pigments which was taken in, and how they have developed that the bigger bacteria have developed the membranous structure. This process is called endosymbiosis. So this is how the cyanobacteria had their difference from the typical eukaryotic eubacterial cell. Thank you. In the next video, we will learn about the structure and reproduction of nostoc.
Want to know more about these topics? Please follow us on Twitter, Facebook, Instagram, and don't forget to subscribe our channel.